want to, of course, thank Nick Hudd and Lauren Williams for inviting me to join everybody in this symposium and talk about some of our recent work. And I also want to thank Nick and Ram for beautifully introducing the subject that I'm going to talk about today, which basically continues on very smoothly from what Ram Krishnamurthy was talking about, which is how you get something as complicated as RNA from presumably rather messy mixtures of prebiotic chemicals. So in the figure that you're seeing in the background here, this is a sort of schematic view of what you think a very, very simple primordial cell or protocell would have looked like, with a simple membrane surrounding small bits of genetic material. And for all the reasons that you've heard about, we think this genetic material is probably RNA or something very much like RNA. And so what I want to talk about today is a little bit about how replication of RNA might have occurred before there were enzymes. And that's an unsolved problem, but we and many other people have been working on it for a long time. So I want to give you a little bit of background about that. And then most of my talk on the same subject that Ram was dealing about. And in this case, it was kind of a surprising outcome that we actually got clues as to how RNA could have emerged from prebiotic mixtures, totally unexpectedly, through our studies of how RNA might have replicated before there were enzymes or even ribozymes around to catalyze that kind of reaction. Okay, so as I said, many people have been looking at this problem of how RNA could have been replicated before there were enzymes for a long time, and the real sort of father of that subfield of prebiotic chemistry is the late Leslie Orgel. And I just want to highlight here one of Leslie's, I think, paramount contributions to the field, which is the recognition that the molecules that are used in biology today, the nucleoside triphosphates shown at the top, are fantastic if you have enzymes that can give you enormous rate acceleration. So having pyrophosphate as a leaving group is something that spontaneously is an extremely slow chemical reaction. And you need something like a highly evolved polymerase to make use of substrates like this. Triphosphates are also fantastic because they're trapped inside the interior of a cell. But in a prebiotic environment, this might not have been such a great thing if you imagine that these substrates for building up polymers were supplied from outside, from environmental prebiotic chemistry. And so the way to get around those problems is to go to molecules like the phosphorimidazoides, such as you see at the bottom here. So in this particular case, the leaving group is this methyl imidazole moiety. And these two methyl imidazoides were invented or discovered, depending on your point of view, by Leslie and his collaborators. And they were studied for many years as a really excellent model system for looking at non-enzymatic RNA copying chemistry. So these kinds of imidazoides are much more reactive spontaneously than triphosphates, and they will start to polymerize spontaneously without an enzyme. You can use just chemistry without any sophisticated kind of catalysis to start building up RNA strands by copying a template. And in addition, you can see that these molecules are much less polar than triphosphates. And so you can at least imagine that these molecules might be constructed on the outside of cells, and yet be able to permeate across primordial membranes, which are inherently more permeable anyways, so they could get to the inside of the cell, take part in copying chemistry and replication, the primordial genetic material. Okay, now methylimidazole, wonderful as it was when it was found, is not the best thing, it turns out. And so in just a couple of years ago, Li, then a postdoc in the lab, did a small screen looking at different potential leaving groups. And he did it in a very systematic way to look at substituents in the imidazole heterocycle at different positions, with different sizes, and with different effects on the pKa. And combining some of the trends that he observed, he then went purposefully to look at this molecule, 2-amino-imidazole, and he made activated nucleotides that look like this 2AI activated version of guanosine, and tested them in copying reactions. And he found that they're actually much more reactive. They're much better at doing copying chemistry than the 2-methyl-imidazole. So I like to say this can give you some idea of the pace of progress in this field, that it only took 20 or 30 years to change a carbon to a nitrogen and make things work better. Now, there's another thing that we like about 2-amino-imidazole, which is that it looks, at least to us, like it's a really potentially prebiotic leaving group, as it can be synthesized in the same reaction mixture as a very related molecule, which I'll talk about much later, 2-amino-oxazole, which is actually a precursor for building up the pyrimidine nucleotides. Okay, so the other major advance that we've made in recent years, it comes from in looking at the detailed mechanism of the copying chemistry. And here the model system that we use is diagrammed on the left. And so we're looking at a kind of very well-controlled, easy to analyze model, which is primer extension. You have a sequence-defined RNA oligomer that can act as a primer when base paired to a longer template. And so the idea is that we want to introduce activated monomers and copy this region of the template by growing the primer strand like this. And for decades, it was thought that this reaction occurred by the direct chemical reaction of the 3' hydroxyl at the end of the primer with the phosphate of an incoming monomer. It turned out that that is not exactly correct. That reaction can happen, but it's incredibly slow. And what actually does happen in solution, and which is by far the predominant way that primer extension proceeds, is that pairs of activated monomers react with each other, as shown on the right, to make a dinucleotide with the structure shown here in the middle. So this is an imidazolium bridged dinucleotide. Very surprising and I think I can say unprecedented dinucleotide structure. But it turns out this is more or less an obligate covalent intermediate in the process of primer extension. So what happens is that these dinucleotides form in solution, or they can also form on the template. They can base pair with the template by 2 Watson-Crick base pairs. And when they do that, this imidazolium bridge, this phosphate-imidazole phosphate bridge, is highly preorganized so as to favor reaction with the 3' hydroxyl at the end of the primer. And as a result, the proximal nucleotide is incorporated into the growing primer chain, and the distal nucleotide is released as an activated monomer so that the cycle can then proceed. We've studied this in considerable detail. both kinetically and also by crystallography. And so what I'm showing you here is an experiment that was done by Wen Zhang when he was a postdoc in the lab. And what Wen had done is he had worked for a long time with a really nice crystallographic model consisting of an RNA duplex with short CC overhangs at each end, to which G monomers can base pair. What he does is to grow these crystals, these RNA crystals, with deoxyguanosine bound to the CC overhang. The CCs would be up above the top of the screen here. He can harvest those crystals, get a lot of them, and then soak the crystals with our activated amino-imidazole activated monomers. 15 minutes after that kind of soaking, the original nucleotides that were here have been displaced. And what you can see in this little snippet of the crystal structure is that we have two activated monomers bound to the template by Watson-Crick base pairing. And I've drawn in the amino-imidazole activating groups, but they're actually totally disordered. They're just flopping around in probably many different conformations. We don't actually see them. We only see the nucleobase and the sugar. But 60 minutes after beginning the soaking experiment, what you can see when you solve the structure of crystals that have been incubated for an hour now is that things have changed. And now we see this imidazolium bridge joining the two monomers that are still Watson-Crick base paired to the template. If you then add a divalent metal ion like magnesium, which is required to catalyze the reaction, and continue the incubation, what you see after 3 hours is that now the monomers have become separated from each other, and there's new electron density in between this monomer and the end of the primer. And so what we've seen in the crystal is the formation of a new phosphodiester bond. So we're seeing primer extension in action through a series of crystal structures. So this is kind of the status of how we understand the primer extension reaction as it happens in a purely all RNA system with an RNA primer template, incoming RNA monomers that go through this covalent intermediate and elongate the primer by forming a new phosphodiester bond. Okay. So, as I just said, that all happens very nicely when everything is RNA and highly purified activated nucleotides, which is obviously not a realistic scenario for anything going on the early Earth. And that concept was mocked, satirized by Joyce and Orgel almost 30 years ago with the phrase the molecular biologist's dream. In other words, this ludicrous idea that there could have been a prebiotic pool somewhere on the early Earth full of beta D ribonucleotides and nothing else. So, of course, we know from all of the work done from many labs that, even though there are pathways emerging that lead to the ribonucleotides, there are going to be other things around. And so that leads to the question that was so nicely addressed in Ram's talk as to how you could get something that looks like modern RNA if there's all this other stuff around at the same time. And so I'll just go over this quickly because Ram, I think, already made this point, but there could be variation at the level of nucleobases. So you could have the canonical A, G, C, and U. You could have variant pyrimidines, such as the 2-thiopyrimidines. You could have variant purines, such as inosine, diaminopurine, and others. You can have variant sugars and linkages. So RNA, we have the canonical 3' to 5' linkage, but of course there can also be 2', 5' linkages. And you can have different sugars like Arabinos, shown here, where the 2' hydroxyl is up above the ring instead of down. You could have sugars like Threos, a 4-carbon sugar that is the building block of a nucleic acid called Threo's nucleic acid, or TNA. first synthesized in the Eschenmoser lab and worked on extensively by many of Eschenmoser's colleagues, and especially by Ram. So if you have all of this heterogeneity, how did we go from a primordial heterogeneous mixture to something that is at least somewhat more homogeneous and therefore likely to function better and be at least a step on the way to the evolution of functional catalysts like ribozymes? Okay, so in this talk I want to focus mostly on two alternate nucleotides that we think would have been likely to be around at the same time as the ribonucleotides, at least to some extent. I'm going to begin by talking about the Arabino nucleotides, where the sugar is arabinose instead of ribose. And so I want to show you the rationale for thinking that Arabino nucleotides could have accompanied prebiotically ribonucleotides. And so what I'm showing you here is a little piece of a pathway towards the pyrimidine nucleotides that's been worked on and developed and optimized in the Sutherland lab in the UK. It has origins itself that go back decades to the work of many other people. But this highly optimized pathway to the ribonucleotides looks, I think, very promising. So the idea is, this is kind of the middle of the pathway, but the idea is you can start with the simplest sugar, the 2-carbon sugar glycolaldehyde, which can react with cyanamide to make this heterocycle, 2-amino-oxazole. As you can see, this is very close to the 2-amino-imidazole heterocycle I was talking about a few minutes ago, which is the activating group for nucleotide polymerization. Here we have oxygen instead of the nitrogen. And this is actually a very interesting molecule because it can be viewed as a kind of storage form, an intermediate that can be built up into a reservoir. It can be purified by sublimation, and it's a nice, stable crystalline molecule. Now, this intermediate in turn can react with the 3-carbon sugar glyceraldehyde to give the following two products: ribose aminoxazoline, shown here, and arabinose aminoxazoline. So the second ring forms either above the sugar, the plane of the sugar, or below. So if it's below, this stereoisomer goes on in a few steps to make ribonucleotides. There's a reaction with cyanoacetylene here to complete the pyrimidine structure, and then 2 further steps to break this bond and give you the desired beta anomer. In this case, the same reaction with cyanoacetylene will complete the pyrimidine ring, and then hydrolysis or thiolysis of this bond will give you the arabinose nucleotides. So you can see they come out of the same pathway. It's very similar chemistry. Very likely that they would both be present. RAO has the nice property of crystallizing out, so it could be built up as a reservoir of this intermediate. But it's possible that most of this could get washed away. But nonetheless, it seems likely that there would be at least some of the arabinonucleotides coming from elaboration of this product. Okay, so just to remind you then, this is what the difference is when you've actually built up the nucleoside. Ribonucleosides, both hydroxyls, 2' and 3' are pointing down below the plane of the sugar. In arabinose, the only difference is that the 2' hydroxyl is above the sugar instead of below. And so the question is, if these kinds of molecules were made together, would they interfere with each other? Can the arabinonucleotides do anything? Would they get in the way of RNA copying? So, what would happen? So what we've done is basically follow the paradigm from Ram's talk, which make these molecules and look at how they react and look at the products and see what comes out of looking at the chemistry. So the first of many experiments that we did to look at this is shown here. Again, we have a primer bound to an RNA template. We have an incoming monomer that in this case is sandwiched in between the primer and a short helper oligo, which is also activated. So these can make the bridged intermediate, which then reacts with the primer. We can make the incoming monomer ribonucleotide or an Arabino nucleotide. And when we do that, we see that typically the Arabino nucleotide is incorporated, leads to primer extension, at a rate that's 10 or 12-fold slower than the corresponding ribonucleotide. Okay, it's slow, but it does happen. When we look then at what would happen next, suppose you get a primer that has been extended with an Arabino nucleotide, then what? Can it keep on growing? And so again, we look at this primer extension using a synthetic primer now ending in either a ribonucleotide or an Arabino nucleotide. And in this case, we have a simplified primer extension system with C's in the template and incoming G's. And now what we see is, kind of to our surprise, is that primer extension with a primer ending in Arabino's nucleotide is very, very slow compared to the primer ending in a normal ribonucleotide. It's so slow that it's actually hard or impossible to get a rate. So it's probably on the order of at least 100-fold slower. So why is that? Every time that we see differences like this, we try to understand what's going on by getting structural information. And so this is another crystal structure from Wen Zhang, using the same kind of model that I showed you before. So we have an RNA duplex with template overhang, incoming activated Gs, but in this case we use an isosteric but unreactive GG dinucleotide. And we used a chemically synthesized primer ending in an Arabino nucleotide. What we see is that the 3' hydroxyl is now much further away from about 2 angstroms further away from the phosphate that it needs to attack in order to grow. And so we think that greater distance goes at least a long way to explaining the very poor reactivity of primers that end in Arabino nucleotides. Interestingly, the 2' hydroxyl is actually positioned pretty well to react, but it appears to be buried within the structure in a way that it makes it inaccessible to the catalytic metal ion. So we don't see any reaction from either hydroxyl. Okay, you can also ask, of course, what happens if there are events that lead to the formation of oligos with internal Arabino nucleotides. Now, these probably can't be made by primer extension, but they could potentially be made in the original set of oligonucleotides that form by non-templated assembly. And here the interesting result is that copying over Arabino nucleotides with RNA actually goes pretty well. So adding a ribonucleotide and copying over an Arabino in the template is almost the same rate, whether the template nucleotide is Arabino or ribo. Okay, so it's not so bad if you find these alternate nucleotides in the template. They can get copied over, but they get copied over preferentially as RNA. Okay, the other variant nucleotide I want to focus on is the Threo nucleotides, the 4-carbon sugars, where the sugar is Threos. And so these are of great interest because they're structurally simpler than RNA, and making them in this pathway is also simpler. So they, again, really look like something that might have been around at the same time as ribonucleotides, which makes it very important to ask, would they be incorporated into RNA during copying chemistry? Would they interfere with copying chemistry? What would happen? Okay, so in terms of the synthesis, the reason it's simpler is, again, we start off with glycolaldehyde reacting with cyanamide to make this 2AO intermediate. Now you don't need to come in with a different sugar. You come in with another glycolaldehyde. You build up the 4-carbon sugar, Threos, in the same form of an amino oxazolane, which gets built out to complete the pyrimidine ring structure down here, and then 2 more steps will make the beta anomer. So how do those react? And basically the conclusion is they're extremely poor. And the reason for that becomes apparent when you look at crystal structures of the reaction intermediates. And so what you're seeing here on the right is the kind of standard RNA situation. Again, this comes from these crystal structures where you have an RNA duplex, a CC template overhang, and here you see this imidazolium-bridged dinucleotide with ribose sugars, so the standard thing. And we have a reasonably good distance and angle of attack of the primer on this adjacent phosphate. Now, if instead we make this bridged intermediate with Threo's nucleotides, such as shown here, what you see is that now the primer 3' hydroxyl distance to this phosphate is much longer, again explaining the very slow rate of reaction. And I think it's intuitively very easy to understand why this problem arises. And that is that in a TNA nucleotide compared to a ribonucleotide, the phosphate is held essentially over the sugar ring, whereas in RNA, it's held away from the sugar ring because of the intervening 5' carbon. So that means that this phosphate, which has to react, has now been pulled away from the primer, and therefore the reaction is extremely poor. Okay. Again, you can put nucleotides in the template. Copying them is not so bad. They're slightly slower, but that reaction actually goes pretty well. So the general trend is that these variant nucleotides react more slowly. If they get incorporated, again, the reaction is slow or sometimes very poor. But when they're in the template, it's not so bad. I want to briefly, let's see. This is, again, trying to understand why this template substitution is not so bad. And so here we're overlaying an RNA oligo structure with the same oligo except with a Threo nucleotide. And what you can see is that the backbone is flexible enough that there's very minimal, entirely local perturbation of the overall structure. So we think this goes a long way to explaining why these nucleotides can be in the template with not too much of a problem. It actually turns out the same thing, the flexibility of the RNA backbone, can explain a couple of other interesting possibilities. I mentioned at the beginning that 2', 5' linkages can form. So especially in untemplated reactions, that result from the spontaneous assembly of nucleotides off the template, turns out there's a lot of these 2', 5' linkages. And so then the question arises, what happens when you try to copy them? And it turns out they can be copied quite well, but when they are copied, the product that is made is a canonical 3', 5' linkage. So again, having this variant structure in the template may slow things down a little bit, but the copying process still works. And what you get out of the copying process is standard RNA. Another thing that we've looked at is pyrophosphate linkages. So it turns out there are some very interesting and simple ways that pyrophosphate linkages can be generated. So, for example, if an RNA chain spontaneously cleaves by intramolecular attack, the 2' hydroxyl, you'll first make a 2' 3' cyclic phosphate that will hydrolyze to give half the time, roughly, a 3' phosphate. That 3' phosphate at the end of a primer actually can be extended, but what you end up with is a pyrophosphate linkage. When templates include pyrophosphate linkages, they actually can be copied over, again generating normal RNA. And the reason why this isn't so bad, again, can be seen from this overlay of crystal structures, that the RNA backbone is flexible enough that the presence of a pyrophosphate instead of a phosphate causes only a minimal and local perturbation of the structure. So all of these experiments together, and others that I haven't had time to talk about, led us to this model. The idea is that prebiotic synthesis would give you a mixture of different kinds of nucleotides. So probably a lot of ribonucleotides, but also likely contaminated with Arabino nucleotides, Threo nucleotides, and also deoxynucleotides, which I also haven't had time to talk about. We think that these different nucleotides would condense in untemplated reactions to generate short oligos that are very messy, very heterogeneous, all these different kinds of backbone chemistries all mixed up together. But every now and then, pairs of these oligos, where the sequence of bases is complementary, would anneal with each other, thus allowing mutually primed RNA synthesis to occur. And even in the presence of a population, a mixture of different activated nucleotides, what we've seen is that the extension, the elongation of the primer, will be primarily from RNA. And so by going through cycles of strands coming apart and back together in different combinations, and each time allowing a little bit of primer extension to occur, we think that what would emerge are populations of oligos that are highly enriched in ribonucleotides. So they would be not entirely pure modern RNA, but much, much closer, much more homogeneous than the original mixture. Okay. This model, of course, makes one wonder how actual RNA replication might have happened. And so I want to end by just very briefly talking about a recent model, which we call the virtual circular genome model, for how actual RNA replication might have occurred, not just copying, but replication. And so the idea is that you could have a population of oligos, all of which map onto a circular sequence, but no, there doesn't have to be any molecule that actually represents this circle. It's just that the whole population of small bits and pieces all map to the circle. And these can come together in all kinds of different combinations to allow ligation or primer extension. And it turns out that in this kind of scenario, it doesn't take very much primer extension to actually allow the whole population to duplicate. So again, we think we have a population of small oligos with different sequences. When conditions are favorable for base pairing, some fraction of the time primer extension will be possible. Then you cycle to, say, a higher temperature or some other condition that allows the small bits and pieces to come apart. Then re-anneal, do a little bit more primer extension. We think that by iterating this process, true replication actually might be possible. So this is one of the main things that we're focusing on in the lab now, and we hope in the coming months or year or two to be able to actually demonstrate not just copying, but full cycles of replication. So if we can do that, the next step would be to actually try to do that inside membrane vesicles, which we already know how to make grow and divide. And so this is kind of the big picture of where this is all heading, at least we hope. towards the laboratory creation of simple protocells that could actually begin to evolve spontaneously. And we hope that by trying to actually demonstrate this in the lab, we'll be able to get some clues as to how this might have actually happened on the early Earth. Okay, I want to end just by thanking some of the people who did this. Much of the work I talked about today was done by Chris Kim when he was a grad student in the lab. The crystal structures were done by Wen Zhang and many other people in the lab, and our collaborator, John Chaput, also contributed to all of these studies. So thank you very much, and I'll stop here and happy to take questions.